Hello.
Hello.
Hi. This is a Science Hangout from Science Mission. I'm Sadashiva Pai. Today we have Dr. Hamid Mirzaei with us. He is the Assistant Professor in the Department of Biochemistry, Center for Alzheimer's and Neurodegenerative Diseases at UT Texas Southwestern Medical Center at Dallas. So Dr. Mirza has won several awards to his credit. As a grad student, he won Outstanding Chemistry Grad Student Award and a Grad Student Teaching Award from Ball State University. He also won Dean's Citation for Academic Excellence. He was the top 10 most cited author of the year by ACS a few years ago. He won Missing Links Award from Cancer Prevention Research Institute of Texas. Welcome, Dr. Mirzaei.
Thank you for having me.
So nice having you here on our Science Hangout. Can you please tell me about yourself, your background, education, etc.?
Sure. I finished my undergraduate studies in Iran. Then I moved to United States in 1999, where I got my master's degree in synthetic organic chemistry from Ball State University. In 2001, I moved to Purdue University to pursue my PhD in the field of bioanalytical chemistry, more specifically proteomics. Proteomics is an area that using mass spectrometry technique to study protein structure, quantity, interactions, and expression. After finishing a brief postdoc at Purdue with my PhD advisor, I moved to Institute for Systems Biology to combine my bioanalytical knowledge with omics and bioinformatics to be able to do systems biology and learn how to use system biology to solve more complex biological problems. Principle of system biology is that instead of using literature or an observation to generate data, to generate the hypotheses, you use omics to generate large data sets as a screen, and then use those data sets to generate hypotheses. And as a result of the nature of the hypothesis generation is not biased. After that, in 2011, I joined the Department of Biochemistry at UT Southwestern in Dallas, Texas. And I have been involved with drug discovery and systems pharmacology by using biochemical tools as well as mass spectrometry.
Can you tell me about your lab, grad student, postdocs, etc. briefly?
Sure. Right now, I have one graduate student, and I work with the senior scientist, more permanent positions. I have two instructors. They're research track faculty. I have a senior research scientist in my lab. I have a senior research associate and a technician. And, I have trained 3 postdocs. They're all having jobs and in making a good living. But currently, I don't have any postdoc, just one graduate student.
Okay. And just going away from research, so other than research, what are your hobbies, like anything you are interested other than research?
Well, actually, this is a very good question. To be honest with you, we are living in a time that we have to spend a significant amount of time understanding the political issues, because as a citizen, I have to vote, and to make a correct decision, I have to be informed, and whatever extra time I have, I spend reading agenda, political agenda, different parties, try to understand what is it that each party is trying to pursue. And I also do the same thing for the local issues in my city to be able to cast my ballot more informed. So essentially, I'm trying to be an informed citizen. And believe it or not, that takes any extra time I have.
Okay. And coming back to the science part of it, like I saw your recent paper on hydralazine, and how did you come across the compound and how that led to all these aging neurodegeneration findings? So can you please elaborate, and you can go ahead and present your work if it is convenient whenever you are ready.
Yeah, I think it's all will be part of my presentation. So I think we're going to go ahead and start the presentation now.
Okay.
Okay, can you see my presentation?
Yep.
Okay, great. I first wanted to say that it took us a little while to build a mass spectrometry facility, and we have a lot of instruments. But in the right corner of my screen, you see faces of very talented and kind and collaborative team of people. I have been very fortunate to be able to work with them, and I learn from them as much as they learn from me. I just want to emphasize that whatever we achieved as a team, and I was blessed with a very good team, very kind, forgiving, and collaborative team. So with that, I would like to start my presentation. Today, I'm going to talk to you about hydralazine, which is an FDA-approved antihypertensive drug, which we recently discovered that might have anti-aging properties. So in this presentation, I am going to tell you that hydralazine has other function than carbonyl chelation, which is a known function of hydralazine. I'm going to show you that hydralazine activates NRF2 pathway, which is a very important pathway for oxidative stress response. I'm going to present data to prove to you that hydralazine indeed activates NRF2 pathway in various cell lines. I'm going to show you that we have validated our finding in cells in C. elegans, which are a family of roundworms. I'm going to present some data showing how hydralazine extends lifespan in C. elegans and keep them healthy, and also at the end present some data that we acquired to show that hydralazine might actually have the potential to help with the symptoms of neurodegenerative disease. So you asked the question, how did you came about hydralazine? So it is well known that neurodegenerative diseases and aging are associated with increased oxidative stress. And that's because many of the defense machineries, such as antioxidant defense machineries and protein folding and other defense pathways, with aging experience a loss of activity. So with elevation of oxidative stress, you expect proteins to become oxidized. So in Alzheimer's disease, there is a protein called tau, and tau in its natural form has a function. It's a tubulin-associated protein, but for some reason that nobody still knows, it assumes a new conformation, which allows it to aggregate and form fibrils and then larger aggregates, which are toxic, neurotoxic, and they kill neurons. nobody knows what causes a molecule of tau that is essentially healthy to go to a seeding tau. And our plan was to use hydralazine as a probe, because hydralazine is FDA approved, so we knew that if we inject it to mice, it's not going to kill them. It's not going to have huge side effects. It's not going to cause oxidative stress. We knew that it crosses the blood-brain barrier, which was very important to us because we wanted to get to those proteins. And we were hoping that if we inject mice with hydralazine and then pull down the proteins that are reacting with hydralazine, we will find interesting protein. So this is the structure of hydralazine, and it has is known to react with carbonyls. So essentially, we wanted to know if we pulled out carbonylated proteins, if we find interesting proteins such as tau or alpha-synuclein or other proteins that are involved in neurodegenerative diseases. And it is known that hydralazine reacts with acrolein, which is a toxin produced after a spinal cord injury. And hydralazine can mediate spinal cord injury by neutralizing acrolein. So we knew that it reacts with carbonyls in vivo. And it had a structure that was not too complex for chemical synthesis. And we also knew that it crosses the blood-brain barrier. So that's, for us, was the key. So the first experiment that we did, we treated cells with hydralazine and then with hydrogen peroxide, which is known to cause oxidative stress, and then hydrogen peroxide and hydralazine. As we expected, the amount of carbonylated protein significantly goes down when we have hydralazine in the presence of hydrogen peroxide. We use a different smaller molecule called hydrazine as a positive control, and we observe the same thing as expected because hydrazine is equally capable of reacting with the carbonyls. So that experiment led to a very important observation. The observation was made by when the data was presented at a group meeting. We realized as a team that hydralazine cells that are treated with hydralazine are not dying when they are treated with hydrogen peroxide. So hydralazine protects cells from hydrogen peroxide-induced cytotoxicity. So we obviously thought, well, it's because it chelates carbonyls and protects the cells. But when we looked at hydrazine, which has the same ability to neutralize carbonyls, we realized that it doesn't protect the cells. So we then realized that there's something else that's happening. So as I mentioned earlier, one of the, my background is in omics, and proteomics is one of the technologies that I'm very well versed at. So we designed an experiment where we grow cells in media that contains heavy isotope-coated amino acids that are chemically the same but distinguishable by mass spectrometry. And then we essentially treat one set of cells with a vehicle or water and another one with hydralazine. We mix them in 1-to-1 ratio, and we measure the proteins. And we look at the proteins that change in ratio. And then we use a software ingenuity pathway analysis, or IPA, to project our data and identify molecular pathways that are activated. So when we did that, we realized that one of the pathways that's activated with hydralazine treatment is NRF2. Now, NRF2 is a very important transcription factor that's upstream of up to 200 antihypertensive oxidative stress functions. They're including thyroidexin and glutathione, NQO1, and many other antioxidant enzymes. So we obviously the stress that we put our cells under was oxidative stress, and it was very interesting to us that the transcription factor that is responsible for responding to that kind of stress gets activated. Of course, in the presence of hydralazine. If you don't have hydralazine, you don't get activation of NRF2. So NRF2 pathway is fairly complicated. It's in a normal condition, NRF2 forms a complex with a protein called KEAP1 and COL3, and it gets ubiquitinated by COL3, and it has a half-life of 30 minutes. And but if there is oxidative stress, then NRF2 is released from KEAP1, and then it gets transported into the nuclei, where it forms a complex with MAF, and then finds the ARE, or antioxidant reactive element, and then expresses all the downstream targets. So we set out to determine if our compound indeed activates NRF2. The first experiment, we look at the expression of NRF2, and as you can see here with Western blot and IP, and as well as mass spectrometry, that treatment of cells with hydralazine in the absence of any stress increases the concentration of NRF2 twofold almost. We also did a reciprocal IP, where in one experiment we pulled down NRF2 and measured KEAP1, and with the other experiment we pulled down KEAP1 and measured NRF2 in the presence of hydralazine. And if hydralazine functions by activating NRF2, it should decrease the interaction between NRF2 and KEAP1. That's exactly what we saw in both IPs. The next step was that, well, if NRF2 is released from its complex, it has to be transported into the nucleus. If it's not in the nucleus, it's not functional. So we looked at the cytosolic level of NRF2 and nucleus level of NRF2, and we determined that level of NRF2 does not change in this cytosol with hydralazine treatment, but it goes up in the nucleus of the cell when it's treated with hydralazine, indicating that not only NRF2 is released from its complex, but it also enters the nucleus of the cell. The next step is that NRF2, according to literature, would get phosphorylated, and before it can form a complex with MAF and bind ARE. So we measured also the phosphorylation of NRF2, both in the cytosol and nucleus, and we show that the nuclear NRF2 phosphorylation significantly increases with hydralazine treatment, but the cytosolic phosphorylation does not change. The next test that we did was to determine if NRF2 indeed binds to ARE. And because if it doesn't bind ARE, it will not be considered functional. So there's an assay where ARE drives the luciferase expression. And in these cells, we showed that when we add NRF2, we can drive luciferase expression in a dose-dependent manner would go up. So it was additional confirmation that binding of NRF2 to ARE's function. We went even further, and we assumed that if NRF2 binds to ARE, the downstream targets of NRF2 should go up with their expression. So we measured the amount of downstream targets of ARE, both by a quantitative PCR and with Western blot, and we showed that all known downstream targets of NRF2 are increased with hydralazine treatment, as one would expect with activation of NRF2 pathway. So although these data are quite convincing that NRF2 is activated with hydralazine treatment, but they were all in vitro data. They are in cells that are cultured and not in a living organism. So to further our study, we used C. elegans, a roundworm, and C. elegans was an ideal model for us because it has a ortholog of NRF2 that we call skin1. And skin1 in C. elegans is expressed in the intestinal nuclei and in the chemosensory neurons in the brain of the animal. So, as a first step, we one that are GFP tagged. That means that if the level of, we could measure a skin1 upregulation by measuring GFP signal. And as you can see in this slide, in the brain of C. elegans treated with hydralazine, obviously there is more expression of skin1. And on the right side of the slide, there is a population analysis showing that the ASI neuron skin1 expression goes up with treatment compatible with our cells. The next, we measured the expression of skin1 in the intestinal nuclei because that's also where this specific transcription factor is expressed. And again, we observed the same thing, that the nuclei with significant number of GFP tagged skin1 go up, and we have upregulation of skin1 in the intestinal nuclei as well. So we wanted to make sure that our findings are correct. So we used a downstream target of a skin1 called GST4 promoter that expresses GFP. So the idea is that if hydralazine activates skin1 in the intestinal nuclei, it should also increase the expression of GFP that is driven by the GST4 promoter. And that's exactly what we see in the left side of the slide, that with hydralazine treatment, worms light up. On the right side, on the other hand, you can see that when a skin1 RNAi is used to knock down skin1, that we no longer see expression of GFP hydralazine treatment, indicating that not only skin1 is activated, but also their downstream targets activated. We also performed a pathway analysis We had done it previously in SHY cells, the neuroblastoma cells, but we hadn't done it in worms. So we repeated the experiment, but instead of using SILAC technology, which is very expensive to do with animals, we used a technology called label-free, and did the same analysis, and we found that NRF2 pathway, or SKIN1 pathway rather, is activated in worms as well. In 2007, Bishop and Garante showed that the SKIN1 that is expressed in ASI neurons mediates the extension of lifespan that is the result of dietary restriction. So it is a well-known fact that dietary restriction or calorie restriction increases the lifespan of animals. And a recent study done at NIH and University of Michigan put this controversy to rest by showing that even in higher primates like chimpanzees, dietary restriction will increase the lifespan. So, but nobody knows how. In this important work by Bishop and Garante, they show that actually it is the SKIN1 that through some endocrine signaling increases the metabolism and leads to the increased longevity. So we thought that if hydralazine indeed activates SKIN1, which what was we observed with our GFP studies, the worms that are treated with hydralazine should experience extension of lifespan, and that's exactly what we observed. That when we treat the worms, their median lifespan increases as much as 25%, which would be equivalent of if a human being living 100 years instead of 75 years. So that's very good, and it shows that hydralazine indeed activates SKIN1 in the chemosensory neurons and extends the lifespan. There are other compounds that are known to increase the lifespan, and such as metformin and curcumin. But an important point in using something like hydralazine is that nobody is going to start taking hydralazine from essentially childhood. And if a drug works, it should work at later stages too. So in this study, we look at late-stage administration of hydralazine, and we actually observed that hydralazine protects the worms up to 17% at the late-stage administration, which is comparable with metformin and is certainly better than curcumin. As a next step, we knocked down SKIN1, and we show that when SKIN1 is knocked down, then hydralazine no longer can increase the lifespan of C. elegans. A major concern with lifespan is the quality of life. Essentially, it means that there is no point in living longer if you're not healthy. So in addition to measuring the lifespan of the worm, we measured their locomotor performance. Locomotor performance of worms is a representative of their health. Essentially, we measure the number of time that they wiggle per minute, and then we use that as a measure of their health. And as you can see in this figure, worms that are treated with hydralazine are showing much lower decrease in their locomotor performance compared to worms in which both isoforms of skin 1 have loss of function mutation. So this proved that not only hydralazine makes the worms live longer, but makes them live healthier too. So we wanted to, there are other mechanisms that could have similar effects. So we looked at, for example, if a hydralazine interferes with the ability of worms to take food, it essentially creates dietary restriction. And so we measured the average pumps per minute and showed that worms that are treated with hydralazine are essentially no different than normal worms in their ability to swallow food. We also looked at other important players known to extend lifespan, such as HIF1 and HSF1, and we showed that none of them change. And it seems that it's specific to hydralazine. We also looked at DAF16, which is also another protein known to increase the lifespan when it's, I believe, downregulated. And we showed that that's also not involved, because when it's mutated and non-functional, we see that still a lifespan is extended without hydralazine treatment. We also looked at bacteria, which are the source of the food for C. elegans. There's a recent study that showed that metformin actually extends the lifespan by changing the metabolism of the bacteria, and that is the reason behind, one of the reasons behind it. In this study, we show that hydralazine does not make any, it does not interfere with the metabolism of bacteria, which would affect the worms as the source of their food. But how about NRF2 in neurodegenerative diseases? It is well known that in neurodegenerative diseases, oxidative stress goes up. Inflammation, infection, they're all causing activation of astroglia, microglia, which are essentially cells that surround the neurons, and they release reactive oxygen species that in turn can damage neurons. But unfortunately, it is scientists have discovered that in many of the diseases such as Alzheimer's disease, Parkinson's disease, and ALS, not only NRF2 is not upregulated to manage the ROS, is actually lower for various reasons. If the level of NRF2 or its downstream targets are significantly decreased, which means that not only cells are under more oxidative stress, their defense mechanisms are become less functional. So we had two questions to answer. We wanted to know if all we observed initially was that cells that are under hydrogen peroxide toxicity experience protection with hydralazine treatment. Hydrogen peroxide is a very loose model for diseases, and this was in vitro. And the other question that we had was that even though hydralazine activates NRF2, but is not tenfold, but rather milder increase in NRF2 activity. And we wanted to know if that level of increase in activity would be enough to protect animals and cells. So in one experiment, we used primary neuronal cells. These are cells that are extracted from the hippocampus of mouse pup at day 0, and they're cultured. And after 20 days, we treated them with rotenone. Rotenone is a mitochondrial complex 1 inhibitor, and mice treated with rotenone develop a Parkinsonian phenotype. So when we treated these primary neuronal cells with rotenone, we see a significant decrease in cell viability. But when we add hydralazine, we almost bring them back to their normal level. Quite good in terms of providing protection. We next focused on our in vivo model, the C. elegans, and treated worms with rotenone again, and we show that hydralazine can also protect C. elegans from rotenone toxicity. We also measured the locomotor performance. Again, there is no point in living longer if you're not healthy. And we are showing here that not only worms treated with rotenone and hydralazine live longer than worms that only are treated with rotenone, but also have better locomotor performance. That means that hydralazine protects both the mechanisms that regulate aging and also mechanisms that underlie the locomotor performance of the worms. We next used a tauopathy model of a C. elegans. These worms have, there are 2 different strains. In one strain, there is a tau expressed that does not aggregate, and the other strain has tau that does aggregate. And here we show that hydralazine protects, increase the lifespan of both, regardless of their the state of tau aggregation, which means that there are very general mechanisms that protect cells. And we observe the same thing with the locomotor performance. So this is essentially a model, a worm model, I should add, for Alzheimer's disease, and we are seeing that the worms are experiencing protection with hydralazine treatment. Finally, we wanted to compare the protective properties of NRF2, and so we compared it with we compared hydralazine with hydrazine, which has the same functional group, with sodium chloride, because hydralazine is a salt, with curcumin and sulforaphane, which sulforaphane is a known NRF2 activator, and it essentially disrupts the interaction between NRF2 and 1. And as you can see, hydralazine provides the best protection of all the other compounds. So I hope that I made the following points in my presentation: that hydralazine protects cells from hydrogen peroxide-induced cell death. Hydralazine does that by activating NRF2 pathway, which is a property in addition to carbonyl chelation. I would like to emphasize that the carbonyl chelation ability of hydralazine is also very helpful in controlling the damage caused by the active oxygen species. Hydralazine extends lifespan and health span in C. elegans, and in a skin 1 dependent manner, which is an ortholog of NRF2, and hydralazine mediated NRF2 skin 1 activation provides more protection. At the end, I wanted to add that Hydralazine is FDA approved, has been in use for many years. All the side effects are known, and it crosses the blood-brain barrier, which is very important when you are trying to find a drug that would treat neurodegenerative diseases. At the end, I would like to thank all of people in my team. All the C. elegans work was done by Ismail Rahman, who's a research associate in my lab, and Mahar Sarami, who's a graduate student who graduated from there. In vitro work, where majority were done by Yichang, Zhang, and Sophie Tu, although some in vitro work was also done with Ismail and Mariam. Proteomics were done by Amir Mansoor Hakimi and Shiva Ramakrishna Yadavani, and the bioinformatics were done by Muhammad Budak. We had a lot of support. UT Southwestern is a highly collaborative environment. My wonderful chair, Dr. Stephen McKnight, provided a lot of support. Dr. Mark Diamond, the director of the Center for Alzheimer's Disease, was very helpful by providing us with the cell models of apathy. Dr. Tambar, who's a star organic chemist, helped us with some organic synthesis. And last but not least, Dr. Ruling Lim and Scott Robertson, who are co-authors of our paper, helped us with all the C. elegans work. We would not have been able to do it without them. Thank you very much for your time, and if there is any question, I'll be happy to answer.
Thank you, Dr. Mirza. You can just, there you go. Thank you very much. It was really very nice presentation. I have a couple of questions.
Sure.
Is the tauopathy in C. elegans, is there any accumulation of tau or anything?
I can barely hear you. Okay, all right. So what was the question again?
In tauopathy in C. elegans, is there any accumulation of tau in the system?
Yes.
In C. elegans?
Yes, exactly. They usually.
Do you see the less amount of tau accumulation?
Yes.
Okay.
I did. There is a lot of data that I have not shown. Obviously, it's not possible to show everything. We have other manuscript in preparation that focus on other aspects of this drug. And yes, we have evidence that hydralazine actually clears these toxic aggregates, but through other mechanisms, which I will not elaborate now, but stay tuned. Look at our future.
Look forward to the paper. Anyways, one more, like, have you tried any mouse model or anything like that for the neurodegenerative diseases?
Sorry, mouse model?
Yeah, any mouse models for tauopathy, like Alzheimer's or anything like that for neurodegeneration?
Yes, we did. You know, these mice, you have to buy them at 2 months of age, and then we have to house them for 6 months before they get old enough and develop the Alzheimer phenotype. And we are in the process of essentially raising these mice to get to the age that they should, and then do the necessary experiments and process the data.
Yeah. I think I don't have anything else other than, like, it's interesting that only one of the proteins was involved. I'm not sure, you might have data for the other proteins involved in this pathway of hydralazine acting on the like neuroprotective effect of hydralazine.
What was the protein?
Is there any other protein? Is the only NRF2 is the only transcription factor that is affected? Nothing else is affecting?
No, there are other pathways that are activated, as I mentioned, but we will
Okay. That is fine.
hopefully publish in near future and other aspects of this drug.
Okay. Yeah, it's interesting. Very nice presentation. You took the point and made the real connection between the hydralazine and NRF2 and the diseases. So thank you very much. I really appreciate. So if you once you are done publishing more papers, well, you can again come back and talk to us about your work. So
absolutely, it was a pleasure, and I hope that work creates some enthusiasm in young people to join and pursue a career in science.
So you don't mind me giving your email address, right? Because I'm going to put it in Facebook and all those things. So your email, if at all anybody is interested in talking to you about any position or anything like that.
Of course.
Okay. Thank you so much again.
Absolutely.